Thank you very much for the invitation to give a talk, and thank you for the introduction. So I'm going to talk about pharmacogenomics and how you can use diagnostics within pharmacogenomics to optimize medicines. Important to remember that medicines are the second biggest budget line for the NHS. In England, we spend about £20 billion a year on medicines, and there's a huge amount of wastage associated with that. So we are all different. We express our individuality by the clothes we wear, by the food we choose, the friends we keep, and the amount of exercise we undertake. But when we look at medicines, clearly we have evidence-based medicine. Evidence-based medicine means that you undertake a randomized controlled trial in a population, show that that medicine works at a population level, and then it's licensed for use in the NHS, and the NICE adopts it. Unfortunately, when I have a patient in my clinic in front of me, I can use that population-based evidence to say, this is the drug I'm going to give to you. And all of us may have a favorite drug, favorite antidepressant, favorite anticoagulant, but that doesn't mean it's going to work in that patient in front of me. So what we tend to do is we tend to use the concept of one drug fits all, one dose fits all in the way we practice. I call that trial and error medicine, unfortunately, because we can't predict the drug's going to work first time. We can't predict who's going to get side effects from the medicine that we give. And that is seen in much of the epidemiological data that's out there. So if you look at efficacy, there is marked variability in efficacy of the drugs that we use. It's been suggested 90% of drugs only work in 30 to 50% of patients. So if you consider an antidepressant, you have to give the first antidepressant, you have to wait 3 months to see whether it works. If that doesn't work, you give another 3 months, and then you give another 3 months. That's not good for the patient, who's not getting any better. It's not good for the healthcare system. On the safety side, we have done some of the biggest studies in the world which show that adverse drug reactions are very important in terms of causing hospital admissions. 6.5% of all admissions to our hospitals are due to adverse drug reactions. At this very moment in time, there are 10 800-bed hospitals occupied by patients with adverse drug reactions. That's 8,000 beds at a cost to NHS England of £2.2 billion a year. When you look at the multi-morbidity population, the elderly population, which is, our population is growing older, the risk of adverse drug reactions is about 15% in terms of causing hospital admission and so on. So it's a huge burden, and we need to be able to improve that. So at the moment, what we do is something that we've done forever, which is kind of empirical intuitive medicine. You make an observation, i.e. a diagnosis. You then prescribe a drug or particular treatment, and you hope you're going to get the right response. It's a trial and error response. And really what we need to do is to go much more towards personalized precision medicine, where you make a diagnosis and then you undertake a test. The test tells you what is going to be the best drug at the best dose in order to get the predictable response. And that's where we want to head to be able to improve what we do with our patients. I'm going to show you some examples where that's already happening, where that's already implemented in the NHS, but also how we need to move to the next stage in terms of what we're trying to do. So the area that I'm going to talk about is pharmacogenetics. The chap on screen is a German pharmacologist called Friedrich Vogel, and he coined the term pharmacogenetics. In 1997, the editor of Nature Biotechnology, Marshall, who's still the editor of Nature Biotechnology, coined the term pharmacogenomics. And the ICH, International Conference on Harmonization, says it's a study of variation in DNA and RNA as related to variation in drug response. I wrote an article in Nature Reviews Genetics last year where I've broadened the definition. I feel that we can use that variation in terms of individualizing dosage and choice of drugs, but actually you can also use genomics and pharmacogenomics to be able to identify new drugs and help in the drug development process. And that's where the pharmaceutical industry is very interested. And in fact, there was a paper in Nature last week which shows that if you use genomic data to be able to characterize your target, you're much more likely to get an effective drug which gets to the market. But I'm just going to focus on the second part of that definition today. So what are the kind of pharmacogenetic testings that we have? Well, you can divide them broadly into reactive and preemptive testing. The current paradigm is reactive testing, that you undertake one of those tests that are on screen when a patient presents to you. You wait for the genetic test result, and then you prescribe the drug accordingly. I think what we need to head to is a preemptive model, where you have a panel test, for example, and in the future exome and whole genome sequencing, that you would then store the data on your electronic health record, and that's available for you when you're seeing the patient, and that's much better and is seamless in terms of your current clinical pathways. Introducing a genetic test and then waiting for test results disrupts your current clinical pathway. And so I think we are going to head towards preemptive testing. I'm going to show you some data and evidence for both of these paradigms. So the first one is point of care, and work I've undertaken, many people in Newcastle have undertaken, including Anne Daly and John Byrne, who was very interested in this, was on warfarin. And I worked with a company called LGC to develop a point-of-care diagnostic. And we did a genome-wide association study which showed there were two particular genes which were important: cytochrome P452C9, which is the enzyme that metabolizes warfarin, and vitamin K peroxidase complex, which is the enzyme inhibited by warfarin. And these are responsible for the majority of dose variation in warfarin individual daily dose requirement. But there are also clinical factors. For example, as you get older, your liver gets smaller, and you require less warfarin. We put that into an algorithm. It was perhaps the first polygenic risk score in terms of pharmacogenomics that was ever created. Once you've got that, you have to show utility. So we did a randomized control trial where we developed that point of care test. In that point of care test, you don't need DNA extraction. It gives you data on 3 alleles in 2 hours. And then we put down an algorithm on the web which told us what the dose should be. And the trial was pharmacogenomic guided dosing versus standard of care. And we were able to show the pharmacogenomic guided dosing was superior to standard dosing. And that was published in the New England Journal of Medicine. So we then went on to revise that point of care test, working with LGC again. And that was 2 hours, the first one in the trial. But we then revised it to get it down to 45 minutes. And you can see the platform there. We then implemented that in a clinic, in clinics in the Northwest, where nurses and pharmacists were doing the clinics. They were able to do the test and then use the algorithm to be able to dose the patients. This again showed equivalent results to the clinical trial in terms of improvement in time and therapeutic range. It was cost effective. The only adverse comment we had from the clinic staff was that because the people had to wait 45 minutes for the test result, it disrupted the clinic. It made the clinic longer. Again, disruption of clinical pathway. Now, there are other point-of-care tests which have been developed for pharmacogenetics. For example, cytochrome P452C19 for clopidogrel use. NICE has done an evaluation of this, particularly in TIA and strokes. And the initial recommendation is that we should be implementing cytochrome P452C9 genotyping for patients who come in with strokes or TIA. Bill Newman in Manchester has done some work on rapid point-of-care genotyping to prevent ototoxicity, deafness in neonates being given gentamicin on the neonatal intensive care unit. And he was able to show that this is feasible to do, and this is also being evaluated by NICE at the moment. So that's a point of care, and I think there's a small number of point of care tests, and you don't need point of care in most cases because you don't need to make that rapid a decision. But we do have other paradigms. So I'm going to tell you about areas that we've been working on, how we've been trying to think about testing in that situation. So I'm interested in patients who get very serious adverse drug reactions, such as the one on screen there. And that is a patient with toxic epidermal necrolysis. I looked after the patient in my hospital. The patient had been given carbamazepine for treatment with epilepsy. 2 weeks later, they developed a rash, which quickly progressed, and they developed blistering of their skin and hemorrhagic mucosal erosions. At the peak of the reaction, 60% of the body surface area was blistered. That's like having third-degree burns affecting 60% of your body surface area. And the mortality from that is 1 in 3. Work that many people have done has shown that the important genes associated with these immune-mediated adverse drug reactions are on the MHC, major histocompatibility complex. There are 3 regions of the HLA, class 1, class 2, and class 3, and all of those may be important for this. And the poster child for this is a drug called abacavir, which is used in HIV. Work done in Australia was able to show association with HLA B5701. Abacavir is associated with hypersensitivity reaction where you get a skin rash, and if you re-challenge a patient with abacavir when they've had a skin rash, they get a much more severe reaction which killed many patients, unfortunately. This is a very strong association. We replicated that in Northwest England, but also did some cost-effectiveness evaluation. And we were able to show in some cases this was cost-dominant, but definitely cost-effective. This data was used by NHS clinics all over the country, and we published this in 2004. By 2006, every patient who was going to get abacavir in this country was having the HLA-B57one genotyping. And the net effect of that has been dramatic. Incidence of abacavir hypersensitivity has gone from 5% to less than 1%. I haven't seen a case of abacavir hypersensitivity for about 7 years now. What the HIV physicians have actually done have gone to preemptive, actually. They don't wait for prescription of abacavir. They actually do 57-1 typing when the patient is first diagnosed with HIV, and they store it on the electronic health record. So they've already done that reactive to preemptive transition. So if you look at the data that's out there on these HLA alleles, at least 30 different HLA alleles have been identified associated with the many different types of adverse drug reactions affecting many different therapeutic moieties, affecting class 1, class 2 alleles, also causing many different types of reactions, skin toxicity, liver injury, bone marrow suppression, muscle toxicity, and all of those are shown on screen there. And so if there are many HLA alleles and so many drugs we're using, can we actually start thinking about using panels for HLA? So work that was undertaken in South Korea looked at data from patients who'd had transplants. And what they did was to identify the particular HLA alleles were relevant to the population, which had been associated with these serious reactions. And then they looked at 12,000 HLA-tested transplant recipients and were able to show 34% had high-risk alleles, 38% were prescribed those risk drugs, and 5% developed these serious adverse drug reactions. And what they suggested was the preemptive HLA information has the potential to prevent serious hypersensitivity reactions. So we've been working with a company called MC Diagnostics, funded by NIH I4I scheme, and we've developed an HLA panel which is able to genotype for multiple HLA alleles. At the moment, if I want an HLA test in my hospital, I have to do a single locus test. It goes away, comes back to me about 2 weeks later. I think the cost, depending on which HLA allele you do, can be between 100 to 250 pounds for that single locus test. We developed an allele panel based on the alleles I showed you in the previous slide, and 85% of the UK population carry at least one of those risk alleles that I showed you on the previous slide. And so the idea here is to move from a reactive to preemptive strategy, whereby you do the test when the patient presents to you, but to store the rest of the data on your electronic health record. This is certainly cheaper than single locus testing. The company has to decide their own price, obviously, but it's less than £50 for the whole panel. Its turnaround time is 48 hours. It's been CE marked and is cost dominant and a health economic analysis. Important thing is also dynamic. You can change the alleles you want on the panel without too much trouble, without increasing the cost associated with that. So let me take you to another one, which I think is a success for the NHS, where we're doing single gene tests but looking at multiple variants on that test. But how do you then say this is enough? How do you then transition to try to improve the testing that you're doing? So this is 5-fluorouracil, but also has some oral analogs such as capecitabine and so on. These are used in 30% of chemotherapy regimens, mostly for GI, breast, other types of cancers. Unfortunately, people can get serious toxicity, bone marrow suppression, GI toxicity, and skin rashes. And the most important element seems to be that patients have a rate-limiting step in terms of metabolizing 5-fluorouracil, and that's the enzyme dihydropyrimidine dehydrogenase, DPYD. And that has polymorphisms in it, and if you have those polymorphisms, then you can't metabolize it and you're more likely to be at risk. So work which has been undertaken, particularly in the Netherlands, has shown that these polymorphisms are very important. I was involved with the European Medicines Agency and sat on one of the specialist advisory groups. That's when they used to talk to us before Brexit. And then chaired a committee for the MHRA. And we looked at the evidence base for this, and we said that we needed to introduce this into the drug label for 5-FU and capecitabine. And that was introduced, and in October 2020, the NHS introduced this test for anybody who's going to get 5-FU. So we went from basically 0 tests to do about 38,000 tests per year, and we're doing 38,000 tests per year now. In England, this is equivalent number proportionally in Scotland, Northern Ireland, and Wales. We've just done a nationwide audit on this, and shows that almost everybody is getting this test, and everybody is getting the test result within 5 days, which is a fantastic achievement. The 4 alleles which have been variants we're looking at are derived from European populations. But it's important to remember that 18% of the UK population is non-white. And so what do we do for those individuals? And are we actually exacerbating health inequalities there? So it's important to be able to then see where we go the next step and how do we actually improve the testing we do for our whole population rather than a sector of our population. So we've done an evaluation of the literature, a systematic analysis, and we identified 53 DPYD variants which have been identified in non-European ancestry populations out there. And if you look at the variants that we're testing for, 3 of those variants are found in other ethnic groups, so that's good in a way. But there's still much more work to be done in those particular ethnic groups to identify other more prevalent variants in those particular ethnic groups. But one variant that we've identified which we don't test for, which is not in the African populations, those alleles are not found that we test for at the moment, is this particular variant, C557A to TG transition, a tyrosine to cysteine substitution, which decreases enzyme function by 40%. The frequency of this is quite high in the African population, relatively, about 2%. And in the States, several labs are testing for this and actually include this variant. At the moment, 4.2% of our population is of black African ancestry. And so we are now pushing the NHS that we should really be introducing this 5th variant into our testing panel so that we're not exacerbating health inequalities. So just to finish off, I want to tell you about preemptive panel testing. And what is the evidence base for that? So I was involved in this particular big trial called PREPARE, which is funded by the European Commission. And this is a cluster randomized implementation study whereby we had 3 countries which started off with pharmacogenomic guided prescribing, and 4 countries, including the UK. Liverpool was the only UK centre which started with standard of care. At 18 months, we swapped over. So it was a crossover design. And we then followed up each patient for at least 3 months to be able to evaluate how they were benefiting, but most importantly, the primary outcome measure was adverse drug reactions and how many were getting adverse drug reactions. And this was the kind of pathway for the patient in terms of the trial. You take a DNA sample for those who were on the test arm and then record it into the electronic medical record. You then give the guidance to the physician, clinician, in terms of what they should be doing with the drug or dose. It wasn't mandatory for the clinician to be able to change the dose or drug. It was up to them. We left it up to them. And then we followed up the patients, as you can see, for those different time points. We also provided a safety code card for the patients who took part in this. We put in the same diagnostic platform in 7 laboratories throughout the EU, which tested for 12 genes, 44 variants, which covers 39 drugs that we use in the NHS as they use in other EU healthcare system. Each patient, as I said, was given this card. It has a QR code, and the patient is able to carry that. And so if they move from one part, if they see different doctors, different pharmacists, different nurses, they can actually scan that QR code. It takes you to a website, tells you what the patient's genotype is, and also then gives you guidance on what you should do if you're going to prescribe a particular drug. So we were doing quite well in terms of recruitment. Our target was 8,000 patients for this particular trial. So we got about 3,581 in time block 1. We were doing well in the second, and we were going to head towards 8,000, and then COVID happened, unfortunately. And so because everything stopped, we had to curtail recruitment, and so we ended up in the end with 6,944 patients. And obviously since then we've been doing various different things, including quality assurance in terms of the adverse drug reaction assessment. So we used a tool called the Liverpool Causality Assessment Tool to be able to look at the adverse drug reactions, and then we had that independently evaluated by the regulatory agency in the Netherlands to make sure that we weren't making the wrong assignments. So this is the data from the Lancet last year, which shows in the primary analysis, which in patients with actionable test results, that the rate of adverse drug reactions was 21% in the test arm, but 27.7% in the control group. And that's a 30% reduction in adverse drug reactions. So for a single intervention, 12-gene panel, which costs less than £100, to be able to produce a 30% reduction in adverse drug reactions is a remarkable finding, actually. And as I said, this wasn't mandatory for people to implement this genetic guided dosing. 70% did, 70% of clinicians did, 30% didn't. The reason 30% didn't was because they didn't have times, they said, to read the guidance that we provided, and others said, we just don't believe in pharmacogenetics, so they didn't implement it. So if they had implemented it, we would have got a better result. So where are we heading for the future? Genomics England is undertaking the newborn screening program, genomes program, where 100,000 newborns will have whole genome genotyping. Obviously, with 100,000 Genomes Project, we've had loads of people who've had whole genome sequencing, and we've got the NHS England program in terms of whole genome sequencing. So lots of people have got pharmacogenomics data already available, and maybe that will need to be extracted and made available in terms of as we move forward into the electronic health record rather than us having to do testing. And so one of the things that we're keen on, obviously, is that we now think that it should be implemented, the kind of panel testing. And I chaired a working party for the Royal College of Physicians, which represents 26 medical specialties, but we also had the Royal College of Psychiatrists there, Royal College of Paediatrics Child Health, Royal College of Anaesthetists, and general practitioners involved in this, and the Royal Pharmaceutical Society as well, working within the Royal College and the British Pharmacological Society, and we highlighted that it was time that we needed to be able to introduce this into the NHS. The major issue for the NHS was going to be diagnostic capacity, lab capacity. If you're going to introduce it for the whole population, 68 million, and there are 250,000 people who go on antidepressant every year, how are you going to do 250,000 extra tests per year? And that's a big challenge for the NHS in terms of introducing this. So I just want to thank people who sort of work with me and our funders, and thank you for your attention.
Thank you very much, Munir. I think that was a wonderful description of some of the challenges that we face, but also some of the real opportunities that there are with the future development of pharmacogenomics. Now, we've got time for one or two quick questions. Anybody got a question for Munir? John.
Thank you. What about the
Yeah, there are some efficacy markers out there, which are not as well worked out as the safety markers. So clopidogrel is an efficacy marker, CYP2C19, for example. And for antidepressants, there are some efficacy markers. But actually, when we look at efficacy, part of the problem seems to be that the effect size is not as large as with the safety. And I think that what we're going to need are probably polygenic risk scores, particularly in in efficacy. How to show the utility of that, we're going to have to do some randomized trials. And as I said, with the polygenic risk score with warfarin, we did a randomized control trial, and that was both efficacy and safety marker we were looking at. So I think we are really going to have to do that in terms of a polygenic risk score. But again, the ability to do that and the cost of that may be prohibitive in terms of doing that.
Okay. Question from John.
By chance yesterday we had a call with George Petrinos about your 44 identified markers, which we can probably now get into a single test. The question is, like with the warfarin test, is it economically viable? If we're going to move at pace to preemptive, then having point-of-care diagnostics probably won't take off. But I mean, my suspicion is it'll be a long time before our electronic patient record is truly preemptive, and so will we still need point-of-care?
Yeah. So the preemptive model, you can have various different models. You can have it at the time, it can be reactive initially, i.e. somebody comes in saying they want to, you want to prescribe something, you do the test, you have the data on that genetic gene that you want, but you also have the additional data you store on your electronic health record. And that's what we recommended in the in the Royal College of Physicians report. In the future, maybe the preemptive could be that you turn 50, or when you have your first NHS health check, that you have your test done. And then obviously the ultimate is the at newborn screening that's going on at the moment. The critical issue, you're quite right, John, is how do you get into the electronic health record? And so we are doing a pilot at the moment in the Northwest as part of the Network of Excellence in Genomics, funded by NHS England, which is looking at how we can actually start integrating this into the electronic health record. What do we need to do with snowbed coding and things like that? Because that's, at the moment, the rate limiting factor is not the diagnostic technologies. It is very much on the integration into the electronic health record system. And by the way, we are looking at the cost effectiveness of that 12 gene panel.
One last question, then we'll have to move on.
Hi, great talk. I just wanted to ask a bit about the clinical adoption part. So you mentioned the paper that you said from the Lancet, and not all of the clinicians adopted that. I was just wondering whether or not the paper that you published would have been sufficient evidence to persuade them to now adopt that, or you still felt that there was resistance?
I think one of the biggest challenges you have with new technologies is changing clinician behavior. I frequently am told that I've been doing this for 30 years, and how dare you come and tell me to do something new. But we can all learn, and we can all try to adopt new things. The reason the HLA-B57 was adopted within 2 years was because it was a new disease. The clinicians were very keen to be able to learn new things and introduce new things for their patients, and there was a very vocal patient lobby. And I think that we are going to eventually get a lot of patient pressure on this. I get a lot of emails, almost every day I get one email asking for pharmacogenomic testing. And patients are getting to know more and more about this, and there's also the legal issues as well. If you look at clopidogrel, in Hawaii, high Polynesian population, the Hawaiian government sued Bristol Myers Squibb and Sanofi Aventis because the label didn't warn them of the fact that the Polynesian population was higher risk of developing lack of efficacy with clopidogrel. They were awarded $858 million by the US courts because of that. So there's the legal issues are also going to play a part as well.
Thanks.
Okay. Thank you very much indeed, Munir.